All right, welcome back to the lecture series in bioinformatics and DNA sequence analysis. So in the part 1, 2, and 3, I took you through the experimental workflow from the colony PCR, running the gel, send it to DNA sequencing, and then look at the sequence file by downloading them and look at the trace file and doing the sequence alignment using BLAST. So you have this result from part 3. Now let's take a look. What we want to do right now is to validate that to validate the identity of our organism. Is it really Staphylococcus edaficus, or is it Staphylococcus saprophyticus, or is it Staphylococcus psilosus, and so on and so forth. After all, their e-value is relatively the same, 0, right? Very, very low. The percent identity is almost the same. The length, the maximum score is similar and all that. So how do we know that your organism is truly Staphylococcus edaficus, for example? Well, in here, we want to look at the MSA. Okay, so you click over here. This is multiple sequence alignment viewer. You click over there. Okay, so what do you see in here, right? So what you see here, so this is the entire length of the sequence. There's from position 0 all the way to position 500. That's the window. You can shorten the window like this. You can lengthen it and all that. So, and then every time you see this red over here, this red here indicate the mismatch. So this is kind of like the map, if you will. So like in the position number 40 or close to 40, maybe 41, 42, near between 40 to 50, there are this mismatch. So the top one right here, it perhaps match best, but toward the end, it's the mismatch getting worse and worse, right? So if you want, what you can do is you can zoom in to this region with a lot of mismatch. So now in here you can't really see the sequence, but if you shorten the window into, let's say, only covering somewhere between about 150. So here, if you align from let's say to 140, yeah, you see the sequence come up. Okay, right there. Okay, so you have the sequence over here. You can just zoom in or zoom out to see your sequence. Okay, so here I'm highlighting only this part between the nucleotide base 40 to perhaps 140 right here. Okay, which is only 100 base pair. So you can see there's this sequence right here that has the mismatch right there. The mismatch is actually indicated by this red letter over here. Okay, so you can see if you look at that, the one at the top, Staphylococcus edaficus right here and Staphylococcus saprophyticus almost match 100%. Look at this, almost match 100%. There's no mismatch. You can actually scroll them all the way, right? Even like if you look at the Staphylococcus psilosus at position number, I don't know, what is this, 158 or so, it's ours is G, but theirs is A. For Staphylococcus psilosus, that's A. Staphylococcus arlete is also A. Hemolyticus is also, this is C, right? So they're this variant, but ours is actually here, G, right? So it's definitely different. It's not A. So we know there's a difference in here. Now, how confident are you that this difference is real? How do you know that, right? How do you know? How sure are you that your letter over here, that this letter, the nucleotide at this position, is truly G? How do you know for sure? Well, in order to answer that question, you need to go back to your trace file. So you can open your trace file again. So here, where's my trace file? Okay, so here, this one at position number, well, remember we trimmed this already in our sequence, so it's really hard to see what they are, but I guess we can do it using brute force. So here, let's say this is what this is a let's find CATTT-GGA. So, and it's close to base number 160. So let's see, go to base 160. This is 100, 110. Let's take a look at 160, close to 160, and we want to find what? CATTT-GG. Where's CATTT? I cannot find it. Where is that? Here. CATTT-GG, close to 160, right there. So and you want to know what you're trying to find out is whether this G actually is real, right? Because other sequence right there for Staphylococcus psilosus, that position has A. For Staphylococcus hemolyticus, that's C, right? And then for Staphylococcus pragensis is T. What about for our Staphylococcus epidermidis? Oh, Staph epidermidis here is actually, there's a difference also in the T right here. So the T, instead of T, the epidermidis has A. If you look at this, in this T position number 156, instead of T, Staph epidermidis has A. So definitely it's a little bit different than your Staph epidermidis. There's A over here, there's a T over there. So let's see whether that sequence is correct. You look at your trace file, that T over here with the middle T of the TTT over here is clearly a T. You can see the peak is very high resolution. There's no confusion in this case. This is really this is real. Okay, that T peak is real in this case. Okay, now after the triple T 1, 2, 3, you have the G. So how real is this G over here? Well, look at the trace. It's really nice single peak. So we know this G is also real, and therefore we know that our organism is definitely different from this other one, including Staphylococcus epidermidis. Okay, so basically that's just how you look at them, right? And here's the catch. Okay, every time there is a difference in the nucleotide, if you see one different in the one nucleotide, it doesn't mean that your organism is not that particular species, but every single mutation, there's a likelihood of mutation, right? If you only see one mutation in the entire sequence, well, you may or you may not. It may be it's just if it's your species, but remember, every single mutation it's rare, right? There's a chance of the mutation for that particular mutation to happen. But when you start seeing multiple mutation right here, for example, the Staphylococcus epidermidis, right, which is what we thought our unknown is, our organism is, right? But you start seeing there are three stretch of mismatch right here. The T over here is mismatched, the A is mismatched, the T over here is mismatched, the A and T over here is also mismatched. When you start seeing multiple mismatch, in fact, not just this space, that one too, the C, G right here, and then it's all the way, it's a lot of mismatch. When you start seeing multiple of these mismatches, then the chance of your organism to be the chance of our organism to be Staphylococcus epidermidis is highly unlikely. Right? It's very, very low, right? So, and it's probably like if you look at Staphylococcus saprophyticus and ataficus all the way till the end, well, saprophyticus, there's one that has this perhaps changes over there, but most of them have no. Oh, there's one here, T. There's one sequence different, the T over here for one of the saprophyticus. So this is another example right here, right? You have 1, 2, 3, 4, 5 Staphylococcus saprophyticus, and some of them have differences. Like this N here, probably because their sequencing is not perfect, right? But there's the Y and the T over here, and there's the one that actually matches ours, which is a C. So you can see that even within the same species, you may have variability, but it's not much, right? It's just 1 out of 500 base pair right here. So again, just because you have one variation in the nucleotide doesn't mean that your organism cannot be that particular species. Again, each mutation, each substitution in particular position is rare, but it's not impossible. But when you start seeing many positions that are mismatched, then you start to question whether your organism really is that species, right? So in here, basically, you see that our sequence, our organism, the one that come from our sequence that come from the organism that we have in the lab, even though we thought it's Staphylococcus epidermidis, it's actually Staphylococcus perhaps saprophyticus or edaphicus. In this particular one, there's no way we can distinguish them, because like in this Staphylococcus right there is very, match 100%, which is the same as Staphylococcus edaphicus. So it's for in this particular one, we cannot resolve that ambiguity. So for all we know, it can be Staphylococcus edaphicus, which is here, or it can be that very specific strain of Staphylococcus saprophyticus right here. Now, what we like to do in the lab is usually to keep documentation, right? So why do we keep documentation? Well, because going through looking at this sequence alignment is not, it's not hard, but it takes time, right? So you want to document them so you don't have to go back through this process over and over and over again every time you have you want to take a look at them, right? So you want to keep all the documentation in one place, and this is what we usually like to do. So in here, basically, I take the snapshot of the multiple sequence alignment, particularly in the region where there are a lot of this variation. So here I take the one with a lot of red cluster over here, starting from the ACA right here. That's your ACA. This is to show the trace file to show that this ACA is actually real. That sequence is real. ACA is not just some noises over there, right? Because if you have question about this GAC, whether the C actually real or is it an A, because other organism has A over here, well, guess what? Just take a look at the sequence file, right? The A, the GAC, the C here is real. It's one single peak, so that way there's no question in terms of the validity of this particular nucleotide identity in that particular position. Okay, so that's basically what we do over here. We like to just see what the variation is. We show the sequence file to make sure that if other professor or other technician want to see, they don't have to call me every time to tell them, yes, this is it, and believe me. They don't have to believe me. They can just look at the documentation, look at for themselves, look at the data, and they can come to the same conclusion, right? Again, they don't have to call me, and they don't have to go through the same process I've done, right? Because it's just gonna be a waste of time. So this documentation is a way for us to save time, to save everybody's time. If there's a new lab tech or a new professor looking into they want to know the identity, they simply can open this file in here, the documentation on that, and just see this is definitely not Staphylococcus epidermidis, because if you look at Staphylococcus epidermidis, actually is really way below that. So here's just the result from BLAST, right? The first couple hit, and then all the way down there, here I've put space over there, all the way down there is the Staph epidermidis, and then after that there's still other Staphylococcus like Staph aureus and stuff like that. Okay, yeah, I think that's pretty much all I want to show you today. So if you have any question about how to interpret the data, if you have any further question about this, or if you're curious about Aspire in general, feel free to ask me. You can either comment down below, just put your comment there, or you can just email me. You know how to contact me. You can just send email to me. Okay, so so I guess that's pretty much all, and I'll see you guys in the next office hour.